Feel free to type in questions. It gives me an idea if I'm way out of bounds in terms of where I'm rambling, and helps to keep me on track. So if you have questions, and then certainly at the intermission about halfway through, as I call it, my theatrical intermission, then we'll be able to answer some further questions as well. Let me see if I can get this one to go down. I'll start up here instead. So just to give you a little bit of an overview, so you know where I'll be talking intro to an MLF in general, and then just giving a few little insights in terms of not just ML, but bringing that border out. So when you're looking at your MLF, malolactic fermentation, it's not just about malolactic. There is a bigger picture to keep in mind. I'll also touch on biogenic amines. I've had a chance to listen to some of Anna Catherine Mansfield's further and Katie Cook's earlier webinars, if some of you have heard those, so hopefully I'll be able to touch in on some of the comments that were brought up during those seminars. How pH and SO2 are related is critical, and I hope to make you more aware of those by the time the webinar has finished. The timing of MLF, it's my little soapbox. I love that topic because I think it just opens so many doors, and the more you understand about it, the more you're able to just read and know about it. Then I think co-inoculation or co-fermentation is a tool to consider. It's not to be used every time. There's some sensory implications that can be positive or negative depending on what your goal is for the wine, but certainly that is something to keep in mind that you could do. Nutrition is certainly something to keep in mind with NL. They're smaller bacteria than yeast. They can't scrounge around as much. And so when things are lacking for them in terms of on a nutritional level, it can become quite critical to get that malolactic fermentation started. And it all ties into, well, if there's not enough nutrition, if the temperature is cold at this time of year. I am from the East Coast, so I do certainly know about cold toes and cold barrels. And then the sensory contribution of not only our bacteria, so not only about the timing as we're talking about in terms of co-inoculation or co-fermentation, but also in terms of different strains having a contribution on a sensory level. Depending on how timing goes, I've got a few other things in terms of Brett management, how your ML can affect keeping Brett at bay, and phenolic compounds and how they affect your malolactic fermentations. Different grapes have different phenolic compositions that can inhibit bacteria as well, so that might be something if the timing allows us. If not, coming soon to a webinar near you, I can bring that topic up again. So the role of microbes. Microbes are important in so many roles in our society when you look at yogurt and cheese and all these other things, but certainly in terms of winemaking, we have conventional, what we're sort of terming, we're using the term conventional yeast, the Saccharomyces species, and that's the ones we're familiar with. It's the fungus, the sugar-loving fungus, as the Saccharomyces means in Latin, and those are the ones we know and are comfortable with in terms of being able to get to the alcohol levels needed in different regions or different sensory contributions. And so that's all part of what the conventional is. But more recently, I think, in the last decade, we've really started to look at what we call the non-conventional, the native emulation. So there's native, where you just let that fermentation go without any inoculation, but that can be risky, mainly in the sense that you can get a great year. You can have a yeast that takes over that's fantastic, that we haven't ever seen before because you're in a different area of the country. But you can also have some pretty scary things happening because that yeast may not be able to complete alcohol beyond 6%, or it may give you some really funky aromas. So that's definitely something that we're looking into. The Torulaspora delbrueckii is one that we've commercialized, and it seems to really be finding a niche for higher sugar fermentations. It's able to tolerate higher osmotic pressure than a Saccharomyces. So if you add the Torula first and then wait for a few degrees, Brix drop, and then add your Saccharomyces, you can take off a lot of pressure on your Saccharomyces, which gives you a tremendous drop in volatile acidity, for example, in a late harvest wine or an ice wine. So there's a lot of, and then different species, they may not even ever ferment, but just their enzyme systems are adding some really interesting aromas. So it's a whole other world that's opening up for us. Bacteria, we've got our The sort of superstar for the bacteria world, whom we're going to talk about primarily today, is the Enococcus eini, or Wienecoccus weenie, as Ralph Kuhnke used to call it. Only if you're planning on an MLF, it's a great strain. As with yeast, we have the Lactobacillus plantarum that's become a great of interest, but we have to work our way through the TTB. I mean, it's a strain that's used in probiotics, in yogurts, etc. But all of a sudden, now that we're adding it to winemaking, it's become something we have to prove that it's okay. But it is a lactic acid bacteria, and it can do a fantastic job with the malolactic conversion. The really interesting part with the Lactobacillus plantarum being that it's a homofermentative strain, which means it doesn't produce volatile acidity even in the presence of sugar. We'll see a little bit more about how the Enococcus works in a few moments, and mold. So there's not a lot to be said with mold. Most of the time you don't want it unless you're looking at a noble rot or a botrytis style wine. So that's just a quick overview of the microbes we're looking at. Not everybody has a microscope. I love to show this picture just because it gives you a bit of an overview of who's the cast of characters in our little scene of fermentation through the weeks once you've harvested. So in the lower right, you've got your Saccharomyces, usually a nice little sphere, and then these little mother cells would bud, and you would have little daughter cells budding off, and that's where that sort of growth of the yeast is very important. Next to that, you have some bad bugs, the Acetobacter, that's the acetic acid that you don't want, and then next to that, you can see some really pretty long chains of Enococcus, and basically they're little round spheres that have all sort of, when they're really healthy, you get these nice long chains forming. So if you're looking under a microscope and you see these long chains of little spheres, you've got a good chance that your ML is about to start or has already started if you're a geek that likes to look under microscopes. Pediococcus is not the greatest bacteria normally to have depending on the species, of course, but Pediococcus is known to produce what they call ropey wines. It produces this yucky, goopy stuff in your wine, and when you pour it, it just kind of goops out of your bottle, which is definitely not something. Some of the lactobacillus are bad, some of them are good. We're discovering some of the good ones, as we just mentioned. And finally, Brettanomyces, or you can see it's got this really interesting sort of more oblong shape. So again, under the microscope is a great way to find out what's going on in your wine. So just two small comments to set the stage, that malolactic is not a separate entity. So often when we get calls, it's like, well, my ML isn't working. I've added it. It's really important to sort of bring that sort of camera, scope it out, and look at the big picture. What happened during your alcoholic fermentation? Did you struggle with your alcoholic fermentation? Was there a temperature spike? Could there have been something going on earlier that would have caused some kind of a stress in that medium, in that wine that was juice, that would affect the ML? Because they are small animals, they would be just as sensitive, if not more sensitive, to something that had happened earlier. So definitely keep that in mind, and I'll give you a tool a little bit later to help that. ML is influenced by the winery and the vineyard practices. So most of the sprays that you might add are designed or are used to not inhibit yeast, because obviously if you had a spray that inhibited yeast, many winemakers would very quickly complain. But not a lot of companies take a lot of interest in what happens with the ML. And we have discovered that some of these sprays have a residual effect in the wine, and can become a problem for the malolactic, and can really cause a delayed or not let the ML go through. And so we've got a couple of different ones that we've isolated, and I'll talk about those in a second. So just going back to this idea that ML is not a separate entity, this is some research that was done at Cornell by Thomas Heine-Klick. This is a few years ago. He's now on the West Coast in the Washington State. But he did this trial where you can see this is nutrients that were added at the beginning of the alcoholic fermentation. So on the side you can see a rehydration nutrient and some complete or mid-fermentation nutrients. So Fermate and DAP are more for the yeast growth, and GoFerm being for the rehydration. But you can see in the red the effect of the ML. This is the malic degradation, and you can see how much more effective it is when you just added a rehydration nutrient to your yeast. So just the fact that the yeast were healthier and protected meant that the ML had an easier time later on degrading that malic acid more efficiently. In fact, the DAP was quite, if you just added DAP, can be almost inhibitory to the malolactic on certain levels. DAP as a nutrient is a sort of a cheap and easy nutrient for yeast, but for the ML, DAP is ineffective. The ML organism is too small to be able to break down the DAP. So just so you have an idea that if you're looking to add quote-unquote nutrients to your ML, DAP is not an option. And this, just to get back to the idea of the spray residues, you can see here that there are the small discs. The color, it's more, look at the disc more than the color for now. And you can see just on the outside of those discs, there's a sort of a space of inhibition, because this whole petri dish is a lawn of bacteria. So it would not have grown if that pesticide inhibited the bacteria just around that disc. So that disc is impregnated with sort of a pesticide residue, and depending on how big that circle is, or that halo around those little discs, it gives you an indication of how much these different pesticides would inhibit bacteria. So it just gives you an idea of something to keep in mind, among the many things to keep in mind. So we have sort of a smaller chart. These are the pesticide names in Europe, but then there's the active ingredient in the middle, and we've over the years figured out the inhibition levels. So some of them are quite high, some of them are quite low. Some bacteria are better resistant to different pesticides as well. So that's a good tool to know if you're sort of scratching your head from one year to the next as to why your ML isn't going through. Maybe have a quick look at what you're doing back in the vineyard. And just to give you this idea, I love this slide because you can fit in over on the upper right-hand side where it says bacteria strain. You can just plug in yeast there because it's the same idea. All these are interrelated and very important, and there's nothing that stands alone in terms of how much it is utilized or how it's being able to function in the grander scheme of a fermentation. So nutritional factors are important, competitive factors from other bacteria, from other yeast, toxic factors, as we just mentioned, in terms of pesticides, cell numbers. You need a critical biomass, a certain amount of living organisms to be in the media to start a fermentation, whether it's yeast or bacteria. So it's important that if you have limiting factors, such as temperature, that it can be something affecting how quickly that your bacteria can get to that, what we call critical biomass, so that they can sort of get the train rolling in terms of getting that ML done. And different juice or must or wine, depending on when you're adding your bacteria, can certainly have an effect as well on your malolactic fermentation. So, the joy of malolactic fermentation. I know people would love it to be like yeast with big bubbles and specific gravities that you can measure, but it is a little bit more of a science. But I think once you see it as your friend rather than your foe, I think it makes it a lot easier. They just discovered some research that if you are inspired to learn a new language, you're going to learn it a lot faster than if you're just sort of being forced to learn a language. So it's a little bit the same idea with your malolactic fermentation. It's a whole world out there of ML, so I hope today I can give you a little bit of insight into that. The chemistry, I think most people understand, is that it's malic being converted to lactic, hence the name malolactic fermentation. But just to keep in mind, there is an isomer. It's a little bit, if you were to cut yourself sort of down the middle, that's a little gruesome, but just this idea that you are the mirror image of that sort of slice down the middle, and it's a little bit like that with isomers of malic acid. There is an L, which is pointing one way, and a D pointing the other way, and the bacteria are able to recognize the difference between these different isomers. So if you're adding some malic acid for whatever reason, I can imagine many of you wanting to add malic because most of the malics are quite high, but just so you know, the commercial editions of the D will remain untouched, so you'll never have that portion of the malic acid converted, utilized by the bacteria, because it is a different isomer. They have a 50-50 isomer blend in these commercial editions. At your next dinner party, you can bring up the fact that Malolactic fermentations are not actually a fermentation. It's really just because no energy is produced. If you think of a yeast fermentation, there's energy, there's heat. That's a true fermentation. It's really just a conversion, which makes it's a small point, but it's good to know, because if you can't, if that enzyme that is converting the malic to lactic isn't able to do that conversion, then that's where it sort of breaks down. Malolactic fermentation can reduce the acidity by 1 to 3, I think actually maybe a bit more, but I should check with my colleague Sibylla. So there is a reduction of acidity, and to quote somebody in one of the previous webinars, the MLF is the only way to preferentially remove malic acid when they were discussing the concept of chemical deacidification. MLF, when we've done trials when comparing chemical deacidification to an MLF, it really makes a difference. You can almost always taste a chemical deacidification, especially if it's done later in the wine making process. So it's a great way to get a biological deacidification happening. Obviously, there's bigger issues when you have very high malic acid, so that's something it's one of the tools perhaps. Production of diacetyl, the buttery aroma, it can be good, it can get bad. If you have a big red wine and you're looking for some of that buttery sort of softness or a little bit of a complexity, that can be a great thing. If you're looking to do a ML on a Riesling, I know somebody, I heard gasps of amazement in the background there, but if you are doing an ML on a Riesling to reduce the acidity, then that's a co-inoculation is a great option, because as we'll see soon, it doesn't affect the sensory level as much. That diacetyl isn't left around to mask some of those beautiful, delicate, for example, Riesling or other white wine aromas. So in general, when you look at the metabolism of our Enococcus ini, what we're looking at is up in the right-hand corner, this malic to lactic conversion, and that's what we sort of see as the malolactic fermentation. These heterofermentative bacteria, which Enococcus is one, as I mentioned before, there are other bacteria that are homofermentative. The reason I'm focusing on this is because, as you can see, heterofermentative organisms do utilize this glucose, and they produce, as you can see down the bottom, this acetic acid. Citric acid can also be utilized and produce the diacetyl and the acetic acid. So there's other factors, other byproducts that we need to keep in mind, and I just wanted, in a very simple way, to illustrate that it isn't just malic and lactic. Your ML is a tool on a sensory level, and the more you know in terms of what else is going on, that's very important as well. But the more you know, the better. And this is what's actually going on in your microbial metabolism and all the other flavor active compounds that are being produced. So it isn't as simple as you might initially think, but it's a lot of this is good, and it's just a matter of knowing the big things that are going on. And what I wanted to do then is to show you this is the big picture. There's a little bit, as you can see at the top, a little bit of sugar being utilized initially and producing your biomass. So that sugar is being utilized by the bacteria to produce more bacteria, and that's an energy source. As you get to that peak biomass, as you can see where that orange biomass line is, you can see that the malic acid is going to be degraded in a perfect world of nice wine. And then as you get to the end of the malic degradation, the bacteria start to utilize the other organic acid, which is the citric acid. And that's towards the end of that, as you see the citric degrading, you can see that's when the acetic acid starts to spike up. Excuse me. So there is this sort of cascade that happens where the bacteria are utilizing the malic, then the citric, and then they'll start to produce acetic acid and go after the sugar again. But we've discovered that we've realized through a lot of research that they don't. If you have bacteria and you have sugar and you have yeast, the bacteria aren't necessarily just going to go after the sugar right away. It's not a time bomb that doesn't have a clock that just sort of, oh, the bacteria are going to go after the sugar. They really do look for malic as an energy source preferentially. So we could see, as I mentioned, a sugar metabolism could only be detected after consumption of the organic acids. So I hope that's clear. What I want to touch in on now is the all-important pH and how if you look at pH 3.5, it's kind of this dividing line between above pH 3.5, there's a greater risk of other spoilage organisms taking over. Enococcus is, it's very, it likes a lot of nutrients. It needs good food, so it's fastidious. And then also it's quite a slow-growing organism. So the more you can do to get that Enococcus sort of taking control first, the better you are, so that you don't get the bad species of Lactobacillus and Pediococcus taking over first. One way that you can do that is by inoculating. Another way is in a lower pH situation. You can, in the lower pH, it inhibits some of these spoilage organisms. So you have a sort of a wider open stage for the Enococcus to have more peace and quiet to build their population and to do the ML. And there's less repercussions because that Enococcus, once it's finished degrading malic and citric, they're not very inspired to go after the sugar in that lower pH. I think they've done what they needed to do in terms of what evolutionary. They're malolactic converters, and in lower pH it sort of protects from further degradation of sugar and spikes in VA. So that sort of range of 3.3 to 3.5 is really ideal. And even there are strains on the market. We have strains that can tolerate 2.93. Getting down to, remember this is a logarithmic scale, so when you're getting below 3, you're getting into some very difficult situations. I mean, if you think of sparkling wine or champagne, they are very often low pH, and we do have sort of guaranteed mLs going through every year, so there are strains that are able to tolerate that, especially if you build them up and treat them like the divas they should be when they're making such great product. In touching on pH, one of the risks of higher pHs is the production of these biogenic amines. So many of you have heard of them in terms of just one of them is a well-known one is histamine. As some of the previous webinars touched on as well, there's putrescine, cadaverine, there's lots of nasty sounding ones, and they are not necessarily in an amount that would make it taste like rotting flesh in your wine, but it is usually in an amount that can cover your very fruity aromas. So if people tell me, oh, I don't like ML because it always makes my wine taste bad, there's probably something else going on, especially if it's a spontaneous fermentation, and also in terms of maybe the pH, etc. So this is just you've got these monoamine oxidases, and these are inhibited by alcohol, which is what would then cause these biogenic amines to form. I've heard being in Australia that the Australians have a reputation for being able to drink, and they also know that they should take their Vegemite, which has a lot of vitamin B in it, to help counter the effects of a lot of drinking. So they were onto something when they, oops, sorry, I spelled vitamin wrong, when they were into this idea that vitamin B helps to act as a cofactor in these enzymes that can then reactivate them quickly and get rid of these biogenic amines, because the biogenic amines, it can cause headaches, it can cause very flushed red cheeks and ears. This is my colleague Sibylla, who's the brainiac of everything fermentation, but especially malolactic, and she has very severe reactions to biogenic amines as she's gotten older and wiser. It's very often women over 40 that can be affected, and hence very often you'll get people who may buy a wine but not buy it again, because if they've had a nasty headache come on, it's not necessarily because of the SO2. If you get people that will often say, you know, I can't drink white wine, red wines because of the SO2, but white wines are okay. That's normally a little red flag that says, well, it's probably not the SO2 that's affecting this person. SO2 reactions are definitely part of some of the population that have an allergy to that, but I think it's a lot smaller than maybe what we think. I think biogenic amines are a bigger cause of issues in terms of headaches coming up with some wines. So just to know that can be a factor when you have an MLF, and something to be aware of if you've had people commenting on that. So you've got our selected strains do not produce these biogenic amines. So, but you never know with the spontaneous bacteria. It depends on who took over, etc. All right. I do want to mention how pH relates to growth. So if you can see here, you've got a residual sugar on one side of 1 gram per liter and a residual sugar of 3 grams per liter. So just showing two different levels, that maybe a higher level would sort of give more volatile acidity. If you look over here on the viable cell counts on the left-hand side, you can see that at these different pHs, pH 3, pH 3.5, 3.3, 3.5, and 3.7, at 3.3 the population diminishes after the ML is finished. You can see over on the end, it's the first one is at the end of MLF, MLF plus 1 month, MLF plus 2 months. So at a lower pH, your population basically dies off once it's finished the malolactic conversion. Whereas at the higher pHs, your ML bacteria are kind of hanging out. They're not really growing, but they're still active. Interestingly, though, that even at the higher sugar levels, the same thing happened. The evolution of the acetic acid, if you relate that back to what we've just seen in terms of population, you can see that the acetic acid level is not really growing once that population has tailed off and is dying off. But you can see that at the higher pHs, that bacteria, because it's still active, is definitely still utilizing what's in the media. And if there's a little bit of residual sugar, it doesn't take much, the bacteria could still possibly produce some volatile acid or acetic acid. Being active, that's what heterofermentative bacteria. So they don't at the higher pHs, they're more comfortable and can still produce more. So it's just important to see how pH can affect your need. This is a quick little chart. It's available to you, and it's again just this idea of having a bigger picture of what's going on in your fermentation. Our big things are alcohol, pH, SO2, and temperature. Those are the 4 big things we would always ask, you know, what's going on with right here and now in your ML in terms of knowing why it may be stuck or sluggish. But also looking at what your yeast nutritional needs. If you had a yeast that was a real nutrient pig, it's probably taken a lot of what the ML might have utilized. The ease of the alcoholic fermentation, how it went through, etc. So this is a chart that just helps you keep the big picture in mind. What the small print was on that was just there's other less understood factors, but things to keep in mind may be dissolved oxygen, the polyphenolic content, the lees compacting. If all your bacteria are sitting in the lees at the bottom, it's going to be very difficult for them to get up into the media and utilize the malolic acid. So keeping that in mind. And as we mentioned, pesticide residues. Really important. I have more slides at the end if we have time, but at the speed I'm going, we might not. But just to keep in mind is how we talk about free SO2 a lot, but really what you want to be considering is your molecular SO2. There are charts available for this. I have a picture of one at the end, so if you want to recognize it. But it's important to know how, if you can see here, at a pH of, say, 3, you just need to add free SO2 of 8.2 to get to that bactericide level of 0.5. That's the sort of important total molecular SO2 that you want to have available. And the molecular SO2 is a relation of pH and SO2 together, and temperature as well in the end. But you can see it at a pH of 3, you need to add 25. When you add 25 free SO2, you're way above what that bacterial side level is. If I have a look at 3.6, if you're at 25 free SO2, you're actually very close to that bactericide level. But then again, if you look at 4, you're way under. So just to show you how pH and SO2 work together. And again, at lower pH, not only are bacteria less active, but you're also going to have a more effective SO2 levels. So that's how pH can be really important. And intermission. There was, I think, some questions I saw back there.
Yeah, we have a couple that came in.
Yep.
One from Todd that really answered to some degree, but we thought we'd have you answer too. Does the use of 71B yeast and its metabolism of malic acid affect the MLF environment?
Okay, so 71B, as far as we know, can degrade probably at the maximum, depending on the conditions, about 30% of the malic acid. So it doesn't degrade all the malic acid. If you have high malic acid, 71B is a great yeast to be part of that team of organisms utilizing, being used to degrade that malic acid. But yeah, 71B doesn't do it all. A lot of and yeast aren't. Most of them, I don't know any yeast that's able to degrade all of the malic. in terms of the Saccharomyces strains. There are a few that do degrade a little bit. So it wouldn't change anything, especially if you have higher malic acids, because then the bacteria would still come in and bring that down, biologically bring down that malic acid.
Okay, and then there was another question that was sent to me privately by mistake, but it was from someone named Tim, and he asks, so taking vitamin B before you drink wine will help counter the biogenic amines?
Yes, it will.
Okay.
I want to be part of that party.
And then there are a couple that just came in now. One from Thomas, who asks, if you co-inoculate, what is the maximum temperature recommended during fermentation? Okay.
Great question. I'll probably get into it a little bit. But so when you're co-inoculating, the bacteria are there from early in the fermentation. Usually within 24 hours you've added your bacteria. Where the peak of the fermentation happens for the yeast, normally it's earlier on in the fermentation, so the alcohol level's not as high, and the bacteria, because they've been in there from the beginning, are able to adapt to that. So the combination of high temperature and alcohol isn't as serious, because your alcohol is usually only about 4 or 5% alcohol in terms of where that peak spike in temperature will occur. So it's not a problem in terms of killing the bacteria. But you don't want to, there's many reasons not to spike a temperature, mainly for the yeast's sake, because if you spike that temperature too high in terms of getting into the 90s, etc., or I don't know if there's any Canadians, but the Celsius, if you go over 30 or 32, you're definitely going to get in. I mean, I wouldn't go over 28 Celsius, just in terms of it will affect the yeast, and it could affect the bacteria as well. But they won't all die right there. You'll get probably a stuck fermentation, because some yeast will survive, but then you won't have enough of a healthy population to finish that fermentation. But in terms of the ml, you should be fine.
Okay, and then there was a second question along with that. Do you recommend adding a mallonutrient at the beginning of a co-inoculation?
No, it's kind of like feeding dogs and cats at the same time. They don't really know the difference between which food is theirs. What I would recommend is adding your mallonutrient once the yeast fermentation is complete, because that's normally when your malolactic fermentation will kick into high gear. The malic bacteria don't really pick up their biomass until the yeast is finished, because the yeast is such a big organism. So yeah, comparatively speaking.
Okay, I think that for now, I'm going to go ahead and hold the rest of the questions till the end.
Okay.
And we'll go ahead and keep going with the presentation.
Okay, sounds good. Oops, did I just jump? Yes. So playing with the time of your MLF addition, there's co-fermentation or co-inoculation. There's different people call them different things. We utilize the term co-inoculation, but and then it's versus the sequential. So depending on which slides I use, I may use the sequential versus co-inoculation. So sequential being you add your ML towards the end or at the very end of your yeast or alcoholic fermentation, which has been very standard. We've got a mentality saying that, you know, if you add yeast and bacteria, you will get huge amounts of volatile acidity. So I'm here to counter that, hopefully. There's other co-fermentations that you could also have, as I mentioned earlier on, where you have 2 yeast being added at the same time or in sequence, very short sequence. So that's why I'm talking about yeast and ML from here on in. Pros and cons. There was an earlier webinar that really had a great slide with pros and cons, and a much more complete one, but this is my little shorter version just for the sake of this presentation, but feel free to refer back to that. Some of the pros are time savings. The fact that your ML might finish a week or 2 after your alcoholic fermentation can make a lot of winemakers in cooler climates, their eyes light up with joy, because the idea that you don't have to nurse a fermentation through a winter and at low SO2 levels, because if you want your ML to go through, you have to stay at low SO2s. Sometimes people are heating their warehouses or bringing tanks into the kitchen. What do I know? But in terms of trying to just get things done more efficiently, a co-inoculation can have a tremendous effect on time savings. There's no alcohol inhibition, because your bacteria are there from the beginning, and they're able to build up with the yeast population. There is a benefit of having that heat temperature from the alcoholic fermentation, especially in cooler climates. You're not heating; it's the yeast doing the work for you to keep those bacteria happy and building a nice biomass. And there's flavor implications that, in my mind, can be very positive, depending on the style you're looking to do. Some of the negatives, you do have to watch for SO2 levels, free and bound. That bound will depending on the pH, become a factor. Yeast compatibility, there is a chart, and I'll show you that in a moment, that we're slowly working through all our yeast to try and figure out which ones are ML friendly versus ML negative, in the sense that there's different factors. Either the yeast produce a lot of SO2, or they're very nutrient demanding and can take away some positive factors for the bacteria to grow. And you do need to be aware of the fact that these heterofermentative organisms, if you're using an Enococcusini, do produce VA from the metabolism of sugars. But that, as I mentioned, is something that you can manage. Just to show you some of the research, this was, if you look at the date, is 1986, so co-inoculation isn't the most newfangled thing we're talking about, but we have brought it back because we know our bacteria better, and we know so much more now that we are able to feel much more confident, because 20 years ago, I believe, it was talked about in the industry, but I think because of the strains we had available at that time and our production methods of these strains, it didn't always work, and sometimes in a very negative way. So I think we know our strains better, what our tolerances for these strains are better in terms of higher alcohols, lower SO2. We understand the metabolism more, so there's a lot to be said for some of the newer research that has come out as well. But you can see in blue, the bacteria in a co-fermentation, co-inoculation, are just sort of hanging out in the background as the yeast population is growing and then slowly tailing off. But it's as they tail off, as they go into their death phase, that the bacteria are picking up steam in terms of building a biomass and getting that ML fermentation going. Some more recent research, this is 2007. This was a Italian researcher, and just to show you, more recent research is coming up with the same idea, this idea that the bacteria are in this sort of darker black squares filled in that says co-inoculo. The co-inoculation is sort of hanging out in the background until the yeast have completed. So it's sort of they are able to, they're not necessarily just sort of rampantly taking off and blocking your yeast and creating VA and causing undue amounts of stress. So they are following their little lifestyle and lifespan in conjunction with the yeast. So one of the main reasons I really like to talk about co-inoculation on a sensory level is this idea that your wines are more fruity. One, because the yeast are still around when the bacteria and the yeast are producing diacetyl, and the yeast are able to reabsorb some of this diacetyl, the buttery aroma that's being produced, so that when you're finished, you have a much lower level of diacetyl, which allows more fruitiness. One of the other things that has come up again and again when we've done trials is that in a more green, vegetative, kind of more, I'm going to use cool climate, but sort of very more green notes in say, like a red wine, we see a lot less of those green vegetative notes when you compare directly the same fruit, same bacteria, same yeast, only the difference in the timing in terms of a co-inoculation versus a sequential. And what we're seeing is that aldehyde is especially responsible for some of these green apple and vegetative sensations. And in a co-inoculation, we have a very early consumption of acetaldehyde. So the peak levels are lower, and therefore, and also maybe other aldehydes and other ketones. So it seems to be that it's related to this as to why the co-inoculation is giving so much more fruity red wines and not overwhelming the white wines with this buttery aroma. So you're getting a degradation in malic acid, a more pleasant acid level in a white wine, and still getting all those nice fruity aromas coming through. High pH reds may or may not, depending on your area, be an issue, but I just wanted to show you, because that would be the ultimate test of a co-inoculation, is if you're looking at pH 3.7, what happens when the other bacteria could possibly take over? First of all, if you're inoculating right away, your population is going to be, your good population is going to be higher than any other nasty bacteria that could be in there and could take over. So you're getting a jump start by having an inoculation. And so if we were to compare just the same, two different yeast, same bacteria, and different times of inoculation, the speed of a co-inoculation in weeks could be actually to end up being months of difference. I mean, you could get a co-inoculation done within 3 weeks after fermentation, or within that sort of couple of weeks after the fermentation, whereas A sequential or a non-inoculated can go on for many, many weeks and turning into months. Just another example in another research station, another area. Lallemont does a lot of research with either universities or research areas around the world. Just the MLF was much faster, and in this case showing that there was not a significant increase in the volatile acidity. So having a co-inoculation didn't necessarily preclude that you were going to have a huge amount of volatile acidity, comparatively speaking. This is the volatile acidity. Sorry, I didn't pull that up. So if we jump into the nutrient formulations for Enococcus, just to give you an idea, it's, as I mentioned, bacteria are a little more tricky, a little bit more finesse is needed to understand them and to appreciate them, because it's hard to know what you need. We don't have a NOPA, we don't have an easy test to find out, oh, this bacteria needs this amount of nutrition like we do for yeast. So you need to really focus on, when you look at that chart that I was showing you earlier, focus on all the positives and make life as comfortable as possible for the bacteria. That can be, if you've never had an issue, not necessarily just mean nutrition. It can just mean in terms of temperature and every other condition, adding the right yeast, etc. But if you've had problems, or if you want to be just on the safe side, nutrition can definitely help. As I mentioned, bacteria is an enzymatic conversion, and manganese is one of those things that's an enzyme cofactor that helps to make the malolactic go. So if you're low on just a small little micronutrient like manganese, that's going to be very hard for that malolactic conversion to get going, that malolactic fermentation. So just that, you need to keep that in mind in terms of what you're doing. Some of the key points are nitrogen. As with yeast, organic nitrogen is the preferential one for, well, yeast don't prefer organic, but it's better for them, much like kids and candy. Their apples is much better for them than a candy, but DAP is a little bit like a candy for a kid. It gives them sugar, but it gives them nothing else, whereas with an apple, you get not only the sugar, but you also get all these other good things, and a slower degradation of that sugar because it's a more complex sugar. So it's a little bit, DAP is a little bit like that. It's a bit of a rush for the yeast and the yeast, but not good for bacteria. The vitamins in these different nutrients that we can provide, and trace elements, manganese, magnesium, and potassium. So as I mentioned before, there are also some inhibitory compounds. Nitrogen, you may think, well, my yeast are going to die and they're going to give some nitrogen as they degrade to the bacteria, but the problem is some of these yeast don't degrade for, say, months after. Like for EC118 is an example where we've seen in a champagne trial, it didn't actually start autolyzing or degrading that cell for 7 to 9 months. So that release of amino acids that will be found in the yeast isn't necessarily going to be available to the bacteria if you're trying to get a bacteria fermentation going sort of a few weeks after alcoholic fermentation. Other things to keep in mind are post-fermentation procedures, clarification, filtration, etc., as to what's going to be available to your bacteria, not only in terms of nutrition, but also in terms of what's going to help it stay afloat and get to those that malic acid that's in the wine or the juice. So that's often why you'll see an ingredient, cellulose, in our nutrients. It's basically just something that the bacteria can attach to. They can go for a little ride around your barrel or your tank. So they don't just all fall to the bottom if there's nothing, if it's a very clear wine at that point. So that's something else to keep in mind. This is this table of compatibility. There's a lot there. I'm just showing you, it's a sort of our working draft of what are the positive ones, D254, W15, our well-known ones, QA23. Some of the ones that are not so good is the V116, produces some SO2, so can inhibit, and as does the WAM. WAM has, it's great if you don't want ML. WAM produces a fair amount of SO2, but not one as available. I'm sorry, and OPAL as well. So and the BM4 by 45 is in that range, and that's because it's a nutrient take. So just as I was mentioning, these different strains have an effect on your ML, and we're realizing that. So just to quickly speed up here, Bacteria need a lot of nutrients. You can see here there's the two first ones are Enococcus ini, the second two are other lactobacilli. You can just see the overall need for nutrition is very high in Enococcus ini, so that's just so you're aware, as I was mentioning earlier. Different strains have different needs for nutrition, so if we were to go through and take out one amino acid at a time and see which one was the limiting one on growth for the bacteria, this strain had 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 amino acids that would inhibit the bacteria if they weren't available to them. And this is the Lalvin 31. It's slower to get started. It's a great strain, tolerates lower pHs, but it is very necessary to have a good nutrition with the Lalvin 31. Compared to, say, another one, for example, alpha, has fewer amino acids that it absolutely needs on an essential level. So it's one that often gets going faster, and that's often reflected as if you look at the nutrient data in terms of how much it's able to get going related to the nutritional level. This is a very easy graph to follow. If you add nutrition, your ML goes through. If you don't, it doesn't. And that's more of a marketing thing. We probably have that on our tech sheet. But it just goes to show that nutrition can be very important depending on the conditions. In this case, alcohol wasn't that high. Total SO2 wasn't that high. The pH is quite nice. But definitely the nutrition helped to get that going in a much more direct fashion. Some of the comparisons, just so you know, a little bit like yeast nutrition, that we do have one that's a rehydration nutrient for the ML, and there's one that's more fermentation. The reason being that ActiML can be added in that rehydration water with the bacteria. Fermentation nutrient, it's more, when we produce it, the pH is quite high. And we've conditioned those bacteria to be, if you're going to be sort of opening the package and just dumping them in, even if adding a little bit of water for 5 minutes would really help the bacteria. But if you do just sort of do that rip and pour or tear, rip and tear and add, the pH is quite different on the OptiMallow Plus. So that's why we recommend adding it to the wine. And therefore, when you add your bacteria, you're not going to sort of swing the pH up and then swing the pH back down as you add it to the wine. So add this one, OptiMallow Plus or OptiMallow, to the wine. ActiML can be added to the rehydration water of the ML. Just to give you an idea, we've done some research. We've realized some of our bacteria have different nutritional needs. So as you can see, as we were talking earlier, the Lalvin 31, it's really important to have good nutrition available, depending on what else is going on, and other ones like the alpha, as you can see on this chart. But this just simplifies it a little bit. All right, sensory. What we did with the sensory, this was one of our sensory projects was with the AWRI, the Australian Wine Research Institute, and this was a 3-year project with, I think it was 3 or 4 different regions, testing the same yeast, the same bacteria, and as we realized later, also readjusting the pH to 3.5 when the tasting went on, because the pH had an effect on the tasting. So everything, whether it's 3.3 or 3.7, got adjusted to 3.5 to do the tasting. Sorry, everything was done in triplicate, so it was a tremendous project. Working with the AWRI is fantastic because they really are the voice of the industry, and the industry listens to them. But it's also like working with Gucci. They're quite they have quite a reputation, and you pay for that. But they're some great people to work with there. This was just to give you an idea how I was mentioning the pH had an effect on the tasting. So you can see with the pH of lower one, you can see there was sort of distinct groups that were forming with these different pHs. So just to be aware of how the pH can affect your tasting of a wine. But basically what I wanted to show you was this, how at the end, the different bacteria came through in very distinct groups of what their effect was in terms of on the wine. So no MLF tend to have more astringent notes, musty or boiled egg sort of components if you did a no MLF or a spontaneous. Whereas different strains would come through with dark berries and spice, chocolate flavors, dark berries in terms of the green 1106. These are just codes for our bacteria strains. Mint and eucalyptus. So, but it was really interesting for us is that what we already knew, we felt, but it was able to be proved over a several years experiment over different regions, Adelaide Hills, Clare Valley, Padaway, and Langhorne Creek, was that these bacteria had an effect on the sensory of the finished wine. So not only is reduction in wine acidity important, but it is much more than just a pH change in the final wine. There are some sensory implications with adding a ML and different strains as well. Diacetyl, just to finish off, in terms of diacetyl, there are different sensory levels in this sense of wine styles. So you can impact of complexity when you're at a lower level. You can have then undesired or desired buttery sensations at very high levels. Different wines have different perception thresholds. So in a Chardonnay, you'll see a much lower perception threshold, and whereas in Cabernet Sauvignon, and different researchers and different books have different thresholds, so it's not an absolute number, but it does give you an idea that in red wines you can take more of this buttery aroma in terms of, or a big red wine like Cabernet Sauvignon style, or a lighter Pinot definitely is more on that level of the white wine in terms of the diacetyl perception threshold. As with our nutrients, we've sort of categorized our bacteria as having different diacetyl concentrations, so this can help guide you in terms of which one you might choose if you're doing a different perception, sensory perception. The beta is a fantastic strain in a co-inoculation, doesn't have a chance to produce a lot of diacetyl. In a sequential, it produces quite a lot, so if you're looking for a big buttery white wine, then the beta's great, but do a sequential. If you're looking for a lot less, it's a great one to choose for fruitiness if you're doing a co-inoculation. Just to keep, this slide will be available to you, so I'm just there's different peaks when that diacetyl happens. So if you days after induction of MLF, you can see that there is an increase in that diacetyl, and it relates to when the citric is being degraded. And then there is a reabsorption of that diacetyl. In beer making, you have a phase called the beer rest, the diacetyl rest after the fermentation, usually a week to 10 days or 2 weeks after the fermentation has completed. You let that beer just sit, because what's happening is that diacetyl is being reabsorbed by the yeast at the bottom, and then eventually you rack off. So it's a very similar idea. That diacetyl can be reabsorbed by the yeast if that yeast is in contact with it. This is looks like a little bit cartoony, but I love this slide. It really shows that anything to do with a longer, slower ML produces higher levels of diacetyl in terms of concentration, and anything with a shorter, faster ML will give you a much fruitier wine. So it's all these different ones. If you have a lower population inoculation, it's going to take you longer to reach that critical biomass. Therefore, you're going to have a longer, slower ML and a higher level of diacetyl. Temperature will also affect how quickly that ML can go through. Aeration, if you're doing a lot of aeration, you're going to have higher levels of diacetyl. So it's a great, easy little slide to post in your lab, and just to remind yourself that that can be a way to change the sensory perception. If you're more of a geek, this is the slide that you want to put up in your lab, or you can have both and compare. But it just shows you that that citric acid, if you're looking down, if you go down, if it's more aerated, if there's more oxygen, you're going to go more towards that diacetyl. If you have more anaerobic conditions, you go straight down and you get to the 2,3-butanediol. They are related, but if the butanediol has a much higher sensory perception, and it gives you the sensation of mouthfeel, but without the diacetyl, the buttery aroma. Diacetyl has a much lower perception, so the butanediol gives you less of that buttery aroma if you're more anaerobic or less oxygen conditions, if you're not doing as much stirring, etc. So that's just to give you the chemistry of it versus the other one. So just quickly in conclusion, I think we've gone through it. The buttery character is something you can manipulate. It doesn't strip. It can add volume. It can add complexity, but not necessarily if you're doing a nice, delicate white wine. So depending on the yeast and the bacteria choice, you can definitely manage what that diacetyl level is. There's lots more to be read about this. We have a whole CD, a whole binder of ML. Chapter 12 is talking about co-inoculation exclusively. It's a really cool topic. I like to try and dispel myths, because it is a tool that you can use, and I hope it's these couple of slides that you've just seen can help at least make you understand that diacetyl isn't always a bad thing. Also, in terms of a sensory, I think. Anyways, you can come back to that. There is a free app, a Lalamon app, so if you want to look up yeast, and even there's a sort of a diagram on how to pick the right bacteria depending on your wine conditions, that's on that app as well, and there's conversions as well. So it's a great little app, and then you can have it all at your fingertips. And Scott Labs has great resources as well on their website. A lot of just sheets if you've got rot and red wines, if you've got cider or something. I mean, they've got a lot of these tech sheets that you can download and print off with really practical information. Anyway, that's me for now.
Okay, Sigrid, great. So there was one question that came in privately that it came to me that I wanted to ask before we get started with the ones that have come in on the list. And it's Steve asking, could juice that has been shipped as part of a wine kit benefit from malolactic fermentation?
A lot of the kits have been adjusted, as far as I know, to sort of not have really high levels of malic. So it would be more in terms of if you're looking to do a buttery Chardonnay, or because there is malic there, it's often either fresh juice or a mix of juice and concentrate, depending on the quality of the kit. But so it would be more of a if you like the style of wine once it's fermented, or if you have the experience to taste through the juice and see that, well, it's pretty balanced already. And I like that fruitiness, then ML might not benefit it. But if you are looking to do some sensory changes in terms of that that more buttery side, then in a kit you could absolutely do an ML.
Okay, and then I had another question that came in just to me from Kurt asking if you had any experience really with the specific cold hardy cultivars, and when you might not use malolactic fermentation.
Some experience, though there's definitely been a lot of evolution. I think I'd need some specific examples, just or sort of names, because there's Treminette, some of the other ones. I'm not thinking properly here. I should pull them up before, but it really depends on if you're trying to emphasize some of that fruit that is in those red or white varietals, and it could be definitely used. It's worth trying. The best thing I can say is to try it, because a lot of these cold varietals, we don't have a lot of experience. Some of the varietals are brand new. You are the winemakers that are going to help us build that sort of repertoire. If you can send us samples for us to taste it at Scott Labs, for example, we can run some samples or some tests on it at the lab as well if we can help. So yeah, we don't have a lot of experience. Some of these varietals are newer, and people may not have the volumes to do a lot of trials, but if you do, it's highly recommended. Do it the way you would normally do it, either whether you're not inoculating or whether you're inoculating after, and do the other one, the other barrel or the other tank. If you're not, tank's probably not possible, but certainly barrel, try and do a co-inoculation. Put that juice in earlier if not possible.
And then Martin asked, is it okay to add Optimalo Plus after MLF has started in the case of a slow fermentation?
Absolutely, because it may be slow because it's lacking nutrient, so yes.
Okay. Frank asked, earlier you mentioned the possible role of phenolics. Are there any implications for our low phenolic wines in cold climates?
So mainly the phenolics aspect was more if it's probably more the high phenolic wines. For example, just because we've done research with it, I'm sorry, I know I should be more relevant to the cooler climate varietals, but Merlot has a very specific phenolic level that seems to, around the world, inhibit ML. It seems to slow ML down somehow or full-on inhibit it. And we've tried to figure out what it is in the ML. Is it, sorry, I'm just going to page through here to the sensory, that's the molecular, come on, sorry. I should probably die. Oh, full screen. Anyways, this is Brett. Here we go. So what we did is we broke down this wine as in tannin extract, anthocyanins, wine extract, grape seed tannins, grape skin tannins. And you could see it had a lot to do with the grape seed tannins that were affecting the inhibition. If you see the level, population level, didn't really increase a lot. And other parts were quite stimulatory. The grape skin tannins were quite stimulatory. So it would be important that regardless of the varietal, I think, is to be aware of grape seed tannins. I think that's probably relevant in many varietals. But certainly there were some varietals that were much more sensitive even with other fractions of the, but we're still doing research on this, so hopefully we can give you more examples of that. I think with the phenolics, it's more, for example, in Pinot Noir, if you, the Pinot needs a little bit of time to stabilize its phenolic structure in terms of the color. So often we'll say, well, it's better to delay your ML in a very lightly structured Pinot because of the phenolics. There's a Pinot and there's a couple of varietals that are quite sensitive to ML because of the pH shift so quickly after the alcoholic fermentation. So it has to do with, but it's certain varietals, Nebbiolo, Pinot. Some of these varietals that we know for a fact are quite sensitive to shifts in pH directly after the alcoholic fermentation. So if you do have a very light phenolic cool climate varietal, then it may be affected by NL on that level, and it may be better to have a sequential and give a couple of weeks to the structure to stabilize the color.
Okay, and then Tim Martinson asked, given the special characteristics of the Minnesota cold hardy cultivars, which is high acid, lots of malic acid, high brix, are there any special considerations in using malolactic fermentation with these cultivars?
Merle would probably be a better one to help chime in on that one, just in the sense that I can see with a higher malic, you may need several different strategies. You may need to do a little bit of chemical early on, chemical deacidification, adding something like the pro-malic, the Schizosaccharomyces pombe strain. It's a yeast strain, but it's been double encapsulated in alginate to make sure that it doesn't escape from the alginate and change, because it can lead to funky aromas if it's a spontaneous Schizosaccharomyces. But it's sort of a combination of deacidification, trying to get a little bit more done with the Schizosaccharomyces, and then finally doing a malolactic. Can be a range of what you might need to do to battle that malic down, because yes, I know malic acids can be 2 or 3 times the level that the noses high in the air Californians tend to think is high. So that's, yeah, my recommendation is to try and look at if you can maybe use several aspects, because only using malolactic, I don't know if they can get through all of that 18 or 20 level of malic acid.
Okay, we're actually past 1 o'clock, but Sigrid, if you're okay continuing to answer questions, we can keep going. Do you have anywhere you need to be?
Nope, I can stay.
Okay, so we'll go ahead and keep answering questions. For those of you who have to go, I did go ahead and post the link to our post-webinar survey, so please go take that if you need to run. But if you want to stick around for questions, then wait, don't click on that link yet. So we have a question from Frank that asks, if cold climate winemakers are planning to build structure using seed tannins, we have to keep an eye out for possible MLS inhibition effects. Was that a question, Frank, or was that more of a comment? I guess.
Probably a bit of both, I would imagine.
Okay, I'll let you comment on that.
Yeah, question. Yeah, I mean, I would think so. I mean, when I see this one, this was research done elsewhere. The seed tannins would probably have an effect on MLF, so probably I would recommend to add them after the MLF is completed.
Okay, and
then I think Corinne has added a comment on the cold hardy cultivars. I think in my mind, I was thinking of sort of upstate New York, et cetera, where they're they've got some of the vinifera varietals, and they have very low color, so that could affect it. But it's interesting to see that the cold hardy cultivars are really quite high on the phenolics. So in that case, I wouldn't hesitate to recommend a co-inoculation on that level, because even if you lose a little bit through a pH shift, you're not going to affect the color to that level.
Okay, and then Tony asks, is there a safe way to halt MLF if a wild one started on accident? Filtration
is probably the best, and adding SO2. Certainly MLF is sensitive to SO2, ML bacteria, so that could be one way to start. And then I think the only way really is, I mean, cold, SO2, cold, and filtration are sort of the three-pronged way to stop an MLF, a wild one, if you really don't want that one to get going, or if you've had bad experiences with a wild or spontaneous ML in your winery and you really wanted to get it started earlier. Another reason to have a co-inoculation, if that is appropriate, Tony, is that the fact that the ML that are getting in there quite early and have time to build that population before the spontaneous would ever even have a chance to get going.
Okay, I think that is all of the questions. If I've missed anyone's question, please retype it really quickly so that we can get it answered. Oh, Richard asks, is it ever too late to start MLS?
It can be, in the sense of what I was sort of mentioning, this that over time there's a clarification that would occur whatever sort of container you've got your wine in. So at a certain point you're going to have a very clarified and very low nutrient possibly situation. So by adding an ML there, you would have to really take into consideration of possibly adding some nutrients and making sure perhaps that the temperature is up, depending on, I mean, if you're starting it in March and it's still quite cold in the cellar, then you're definitely going to see an effect on how long ago you had any yeast in that area. The yeast might add some nutrition over the time, but if you've gotten it off lees, all these kind of, there's a lot of factors, as I say. It's not just, you can definitely get it going if the malic is there and if you've got some good nutrition and the temperature's right. And some people do that. I mean, they get it cold enough that there's no chance of it starting through the winter and then just deal with it in the spring. So, but yeah, make sure you keep an eye on that nutrition and having some kind of particulate matter around for the yeast, the bacteria to grab onto.
Okay, I think that's all the questions. So Sigrid, thank you so very much for the great presentation. I think that everybody learned a lot and really enjoyed having you. I know I was glad to have some industry participation. This has been great.
It's been a huge pleasure. Thank you very much.
All right.
It was my first webinar, so I'm glad I survived, and I'm glad everybody else survived.
Yeah, and then also just a few final notes. The link to the post-webinar survey, I'm going to throw that right back up there. If you would please go take that, that really helps us improve our webinars as the project progresses. Another note is the upcoming webinars for the 2013 series are up there on the screen now. One thing that I forgot to add is the February 12th webinar, Justine Van Heuvel from Cornell is going to be joining Harleen Hatterman-Valenti for that session. And with that, thanks again, Sigrid. Thanks, Merle, for being on hand to answer questions. Thanks to everyone who participated in the webinar today. It's really nice to get such great participation from that. I see we have a question about the PDFs of the slides. Yes, they are available on our project website on the Recorded Webinars tab. I went ahead and threw those up earlier today, but didn't have a chance to get an email out to everyone. So yes, the PDF version of the slides is up. Thank you, Sigrid, for allowing us to do that. We really appreciate that as well.
Thank you.
So, all right. With that, I think we're done. And thanks a lot. So, and Sigrid, I'll talk to you again this evening.
Sounds good. See ya. Bye.
All right. Thanks. Bye.